Hello everyone, till now we have studied and understood about the semi-conservative nature of DNA. And now two things come in our mind when we think about replication of DNA. First is autocatalysis. Autocatalysis is a condition in which we see that the DNA is itself replicating itself, or it is self-replicating itself. So from a DNA is formed. That is normal process of DNA replication, which basically occurs. And in by this category or by this method, a DNA is capable of forming replica of itself. Watson and Crick were the first people who identified this characteristic of replication being present in the DNA when they defined the double helical structure of DNA, because they were aware that the nucleotides, they have complementary base pairing, and when we unwind the DNA or we separate both the strands, each of the strand they have the capability of forming replica of their own strand. So by this process he got an idea that it is very easily possible to form a DNA from a same DNA. Just the thing will be there that it will be maintaining the semi-conservative nature, and one of the DNA molecule will be a parent, whereas the new one which will be formed will be a daughter DNA. But still the expression or the behavior of both the new strands which are formed will be exactly like the previous parental strands. So in this way, the autocatalytic mode of replication was understood. And if we want to concise the entire information, just means that DNA is itself capable of forming DNA, can form DNA. And how can it form that? By the process of self replication. Next is like how this process is actually taking place. Just see this entire structure of DNA. If you see the structure, this is a double helical structure which is visible over here, and just both the strands they have got separated. After getting separated, each of the strand they that is forming a replica of itself, and this is known as autocatalytic mode. And how can it form? It can form in this way because of the complementary base pairing of the nucleotides. A will always pair with T and C will always pair with G. Keeping this nucleotide complementary base pairing in mind every time there is formation of a new strand. Now, as autocatalysis is there, similarly there is another term which is heterocatalysis. Here, the understanding is little slightly different from the autocatalytic mode. In the autocatalytic mode, DNA to DNA formation was taking place, but here DNA to RNA formation takes place. It means that whenever there is a requirement of a particular protein or an enzyme, we are aware that DNA is the main gene from where a particular protein is formed or an enzyme is formed. So whenever our body lacks a particular DNA or particular enzyme, so suppose in the DNA segment, one of the portion is there which is capable of forming an enzyme, and that particular enzyme, and that enzyme is lacking in my body. So my body will trigger this portion of DNA, and segment of gene will be activated, and it will form the particular enzyme which is required, and my body will get the enzyme. And for this process, heterocatalysis occurs. That is the conversion of DNA to mRNA and further mRNA to protein and formation of enzyme. Okay, let's see how does it occur. So when our body determines that it requires more of something, a protein, or in a cell, the enzyme is secreted into the cell's nucleus, and this which causes the DNA molecule to open up at a specified place. Why a specified place? Because that small portion of, or this small segment of DNA will only be capable of forming the enzyme which is required by us. So that requirement will be fulfilled by forming a mRNA. This mRNA will have purines and pyrimidines in the similar way, but it is an RNA, so uracil will be there in spite of thymine. That is the only change, and other than that it will be the same messenger RNA. From the nucleus, the information will go on to the cytoplasm. In the cytoplasm, proteins will be formed, and that protein through the Golgi bodies will go out of the cell, and enzyme carrying the enzyme which is formed and trigger the organ where the requirement is. And this is how heterocatalysis is helping us in getting the required enzyme or protein. So the difference between autocatalysis and heterocatalysis is during the process of autocatalysis, DNA to DNA formation takes place. That is duplication of DNA, which occurs in the S phase or synthesis phase of cell cycle occurs. Whereas in heterocatalytic mode, DNA will be there as such, but from the DNA, one small segment will be taken into consideration, and that segment will produce an mRNA, which will have the similar complementary base pairing like the parental DNA, but only difference will be thymine will be replaced by uracil because it is a, yes, RNA. And as this RNA moves on to another portion, it will form a protein. After protein formation, it will form the enzyme, and the activity of the enzyme will be seen in the organ. This is about autocatalytic or heterocatalytic mode of replication. Now, we will be discussing in detail about DNA replication, and for that we need some enzymes. For the replication process of the DNA, certain enzymes are required, and information about each of the enzyme is very important. The first enzyme which is required, it is not first, second or third, it is just I have arranged them in a certain category so that it is easy for remembering. First enzyme which we think of is the helicase. As the name is, helicase, so it is capable of unwinding the coil of DNA by breaking the hydrogen bonds. So if you think of these structure of double helical structure of DNA, this opening up will take place of the DNA with the help of helicase, and helicase will open the DNA by breaking down the hydrogen bonds. There are two kinds of twists when we think about this DNA structure, and that's why we need two different kinds of enzyme. One of is helicase, which will have help in opening up. Now think about a rope. When we try to like think two ropes being arranged the way a DNA is arranged in a double helical form. One is a circular twist and another one is just a twist in itself. We can open the twist in itself like the way I have tried to open it. But what about the curves, the circular curves it has? So in order to get it rid of this circular curve over here, we need SSB, which is single stranded stabilizing enzyme or single stranded stabilizing protein. This protein helps in providing strength. Like what it exactly does is it incorporates the position in the DNA strand. It first of all cuts the strand over here, circulates it, and circular twisting will be there, and then again this strand will be binded to the same strand, and with the help of this the tension in the strand is reduced. So it provides strength to the DNA structure, and it helps in stabilizing the circular twist of the DNA. These are globular proteins. They will just come and bind up over here so that the circular twist is not seen, and uncoiling doesn't, again, recoiling doesn't take place. After this, there is another enzyme which is topoisomerase. Topoisomerase is an enzyme which is utilized, or the function of it is that it removes the tension of the coil. So by cutting the enzyme into two portions, or not the enzyme, rather it helps in cutting both the strands and rotating it and again resealing it with the DNA so that the tension is released, and that is about the topoisomerase. Ligase helps in binding of the cut off DNA. DNA polymerase 1, it helps in removal of primer and filling the gap. Primers are small RNA, okay, we'll study about them. Primers are small 10 to 12 nucleotide RNA structures which help in forming the entire DNA strand. Next series of enzyme is a primase, um, which is helpful in synthesis of the primer. I said that primer was a 10 to 12 strand RNA structure, or a strand we can say. So primase, it helps in primer synthesis, and it is a DNA dependent RNA polymerase. It is dependent on a DNA for the formation of RNA strand. DNA polymerase 3 is the next enzyme, which means that it has several subunits at first. The main function is that it helps in the synthesis of DNA, but the several subunits it has, let's just have little idea about them. Alpha subunit, it provides catalytic bound, or it has a catalytic property. or catalytic bonding. Catalytic effect means it has in breaking down. Okay, beta 2 structure is there, which is a clamp, sliding clamp, in the sense whenever the DNA synthesis, DNA synthesis, or you can say that replication is occurring, at that time this entire DNA polymerase has to slide over a structure. So this sliding movement can be initiated with the help of beta 2 clamp. It helps in clamping or helps in fixing the DNA polymerase chain or polymerase subunits together into this strand. The orange one is the strand, and the strand will be fixed into that place for the synthesis. Epsilon subunit is also present in the same uh, DNA polymerase tree structure, and it helps in proofreading. This entire DNA polymerase is actually a dimer, and it helps in synthesis of both the strands at the same time. So you can think of like two DNA strands, like one of the DNAs, this is the DNA. When it opens up, both the strands are synthesized at the same time. It is not that only one of the strand will be synthesized, and clamp will be attached to a place which is like somewhat in the middle. One side will be having the DNA polymerase 3, another side will also have DNA polymerase 3. I've just drawn one of the structure, but both these structures, they will slide, and as they are sliding, music is taking place. We'll just look up to the structure in details after some time. Next is a phosphorylase. It helps in addition of phosphorus, or it helps in activation of the phosphorus. Nuclease is an enzyme which helps in proofreading, or not only that, sometimes even cutting of the nucleotides is done with the help of nucleases. Activated nucleotides are required for the process of DNA replication, like AMP. Suppose AMP is what? Adenosine monophosphate. It gets phosphorylated to become adenosine triphosphate, and it provides energy for the process of replication to occur. And not only that, it is not only adenosine monophosphate, TMP is there, CMP is there, GMP is there. All of them, they form their triphosphate structures. So that three, they become little more activated, and with the help of more activation, they can form or produce a lot of energy, which is utilized for the process of DNA replication. Now you all can see this structure. Just let's recapture the entire things which we have tried to understand. We will start with the parent DNA. This is the parent DNA, and it has just uncoiled with the help of topoisomerase. What is the function of topoisomerase? It helps in decreasing the tension among the double helical structure. In the sense, if think about two coil structures, if we uncoil them, then they again have a tendency to recoil themselves. So if a band is attached at this position, this will help in reducing the tension and not at all allowing them to again recoil, and that is the help of or that is the function of topoisomerase. You can see these four dots. These all are single stranded DNA binding proteins, or SSBs. Their function is to stabilize the enzyme. Now the stabilization of the enzyme helps in even giving them strength. The strength is provided to the DNA structure in order to be stabilized, and no recoiling is allowed during this process. Then after, there is addition of this RNA. I should say about helicase at first. Helicase will help in uncoiling the DNA. RNA primer comes and gets attached at this place. RNA primer is 6 to 7 or RNA primase is actually an enzyme basically, which helps in formation of a RNA primer. So this primer red color structure is actually RNA containing, or it has RNA. It means that it has presence of uracil in spite of thymine. So this entire structure, which is red color, will be removed later on, which is named as RNA primer once the DNA is completely formed. You can see the structure of DNA polymerase 3 on the leading strand. Again, leading and lagging strand, two terms are used. DNA polymerase 3 has a dimer activity. It means both, like two DNA polymerases are present at the same time. The way it is shown over here, it is not exactly this way. Both of them, they are like DNA polymerases. They are together moving in five, like in one direction, and they form, like they move once suppose one of them is moving in this way, this is the actual plane on which both the dimers should be moving. If they move in 5 prime to 3 prime direction, the for one of the strand it is right, or the movement is correct, but whereas for the another strand it is opposite. But still the formation of primer will be taking place in a discontinuous way in one of the, or formation of DNA synthesis will be occurring in one of the strand which is 3 prime to 5 prime in a discontinuous way, whereas 5 prime to 3 prime will be formed in a continuous way. When it is a continuous strand, it is known as a leading strand, whereas the strand or DNA strand which is newly formed is discontinuously formed, it is known as a lagging strand. And as this dimer is moving, it forms the primer in both. Suppose in case of this lagging strand, the primers are formed several times, because every time the formation of the DNA is always in 5 prime to 3 prime direction. So from 5 prime to 3 prime there will be formation of this small strand of DNA, again small strand will be formed, again small strand. So these all will be joined later on with the help of ligase, and again the polymerase, DNA polymerase will be running over it, so that removal of the RNA primer takes place and formation of actual DNA strands occur in the entire Okazaki fragment. As because this fragment or lagging strand was found out by a scientist named as Okazaki, so the fragments or these small portions of the RNA or DNA fragments which are formed containing some of the portions of RNA. See this green portion which I have made, this is an RNA, 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 and then there is synthesis of DNA. This is a little idea about all the enzymes and how do these all enzymes function. The function of each of the enzyme is to be learned properly.